X-Talks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The X-Talks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the X-Talks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at xtalks.com. And this week, I am happy to introduce two very special guests on the show. We have with us today Dr. Howard McLeod, who is the Director of the Center for Precision Medicine and Functional Genomics and Professor of Pharmacy and Medicine at Utah Tech University, as well as the Precision Medicine Advisor at the Geriatric Oncology Consortium. We also have Dr. Sharmeen Roy, who's the Chief Strategy and Science Officer at DoseMe, which is the world's first and largest Bayesian dosing platform designed for clinical practice. Dr. Howard McLeod is an internationally recognized expert in precision medicine who has made novel contributions at the discovery, translation, implementation, and policy levels. Dr. McLeod received his doctorate in pharmacy from the Philadelphia College of Pharmacy and Science and did his postdoctoral training at St. Jude's Children's Research Hospital and the University of Glasgow. Dr. Sharmeen Roy is passionate about leveraging technology to amplify the impact of the pharmacist. Her career spans pediatric clinical pharmacy, clinical research, and pharmacogenomics with leadership roles at University of Chicago Medical Center and Pipeline RX. She received her Doctor of Pharmacy degree from the University of Illinois at Chicago, is a board-certified pharmacotherapy specialist, and completed a pediatric specialty pharmacotherapy residency at Texas Children's Hospital. To learn more about the current landscape of precision medicine, including precision and optimized dosing, let's turn to Dr. McLeod and Dr. Roy now. Thank you both for being on the show today.
Thank you, Ayesha, for having us.
So the first question I'd like to start off by asking, and I think I'd like to get both of your insights on this actually, is what does precision medicine mean in today's drug development and clinical landscape? And how has the field evolved over, let's say, the past decade? And where do you see it going?
Maybe I'll jump in first. I think precision medicine is an aspirational goal. We want to be better, smarter, more precise than we used to be. And so in some ways, we used to be more precise because we would use different dosing forms 30 years ago that we don't even have now. But now with the current forms we have, the current regulatory structure, how can we be better? How can we choose a drug more appropriately? And I think that precision medicine to some people means a particular type of assay or particular type of matrix, plasma or DNA, et cetera. But to me, it's really, can we better ourselves? Can we go from good to great to, heaven forbid, perfect? And that's the ultimate goal. May not be achieved, but we need to at least be shooting for that.
I'd like to echo that. I'm a pediatric trained pharmacist, so I always tell people that I've been doing precision medicine for a very long time because every child is different. The ages vary, right? From day zero to 18-year-old, their dosing is very different. So, I think taking that and just expanding it to all patients, I think we want to, like Howard said, we want to be better than we were before. And I think with the current, amount of data and information that's available, we want to take that and leverage it to do better than we did before as far as taking care of patients and really ultimately always keeping the patients at the center of whatever we do.
Absolutely. And Dr. Roy, I'd like to ask you more about DoseMe and how that came about and what it's all about.
Sure. So DoseMe is a precision medicine platform. Now like we talk about mentioning precision medicine in many, many different aspects. This is just one aspect of it, and this is the dosing aspect. So, we take dosing that is approved by the FDA and label on the label, and you really essentially take information that's available to you through population data and individualize it for our patients. So the software, what it allows is it takes complex models of drug dosing and helps you take that and integrate it with the patient data and really individualize and make sure that you're dosing for that specific patient versus a larger patient population.
And Dr. McLeod, what you've been in the field for a long time with a very illustrious career, and so how have you seen things evolve over your time in the field? And what brought you to precision medicine, and what keeps driving you in the field?
Yeah, thank you for not saying how many years I've been active in the field. I appreciate that. I think when I first trained, I, like most of my cohort, were one size fits all type of people. We didn't know any better. And then as we get out into practice and research and start understanding how much variation there is amongst people, even if they're the same size and have as much similarity as possible, you start trying to figure out, well, why is that the case, and how can we measure it? And my first exposure was using blood levels to guide therapy, cancer therapy, anti-infective therapy, immunosuppressive therapy, lots of different options that we were working on. And then as time went on, we started adding additional things. So with blood levels, one can only look once you've given the drug. With DNA analysis, one can start doing that before you ever give a drug. And so we started adding that. And I think it's really addition. DNA followed by blood levels are, to me, a double safety approach to really try to get the right dose. And so that was really and it was patience. We had a little girl who nearly died under my care at St. Jude's. And as we tried to figure out why a normal dose for everyone else was an overdose for her, we ended up discovering there was a gene variant that caused her not to be able to break down the medicine. And so that was the first part that started dragging me into this field of, why is there differences? Can we understand the mechanism? Can we then apply it to routine care? And that's been really what's gone on over the years.
And so you mentioned new technologies like DNA technologies now being combined with blood testing. And so would you say that's been sort of a big game changer in precision medicine? And what other technologies have had kind of a similar impact, or maybe future technologies that you're looking forward to?
Yeah. So technology, right now, we're not getting the most out of the technologies we have. And that's it's where the science meets the psychology, if you will. It's the idea that one would identify risk with DNA and then fine tune things with blood levels is normal if you're trying to manage a blood thinner like warfarin, where you can use international normalized ratio as a measure of clotting and try to know. But when you get into some other areas, especially toxic drugs like cancer chemotherapy, it's as if there's this foreign concept that no one can understand. And so the same patient may have individualized blood level guided vancomycin therapy or other anti-infectives, but then the idea that we would also standardize their cancer drug, it's as if we were saying something, that a word that was the first time it had ever been spoken in mankind, and so there's a lot of work to be done to help people realize we're already doing that to some extent. We need to just start applying it more broadly. And so where blood level guided therapy is being applied is wonderful. It's a real plus, but there's so much more opportunity, and it's not just going to be science. It's going to be the really convincing people that you do not need to be scared, that giving the person the right dose is a good thing. Because if you've grown up giving 200 milligrams of a drug, the idea that someone needs 100 milligrams and someone needs 300 milligrams is very scary, and we just need to normalize that. And so I think the technology helps. New technologies are exciting, but we also have some good old-fashioned clinical management to grow.
Those are excellent insights, and that speaks to what you're doing at DoseMe, doesn't it, Dr. Roy? And so how do these technologies and how do these concepts of precision medicine, number one, help pharmacists, of course, in better establishing and optimizing dosing, but also how does that translate to the patient, and what is the patient reception, as Dr. McLeod alluded to, where there might be hesitancy when they hear a word like precision medicine, and why am I not getting the same dose as another person or another patient? And so how does all of that pan out and translate?
Yeah. So I think I touched on this a little bit, where our software takes complex math essentially and simplifies it. So then the clinician at the bedside, the pharmacist at the bedside can use it at the bedside for the patient and spend that time individualizing dose for the patient rather than spending time trying to figure out what that individual dose may be. So, it's the platform takes a lot of information from these population data. It takes information from the patient's individual data and helps the clinician, kind of deliver that dose, the appropriate dose for the patient. And really, I think the key to any successful technology implementation is you have to make it easy for the pharmacist or the clinician, and you have to make sure you're not too interrupting their workflow too much. Because if you can accomplish those two things, then the adoption just, increases tremendously. And so that's what we are constantly trying to optimize, is making sure that, it fits into the patient care that they're providing at the bedside. And it's not, asking the clinician to step aside and focus on something that is other than the patient. And so I think we just need to always think of technology as something that's an asset. We're going to use it to make us provide, as clinicians, better patient care versus, it being kind of a hindrance. So, that's kind of what we have as far as in our mindset when we're looking at how it's going to help a pharmacist or a clinician that's doing dosing, or a clinical pharmacologist, whoever the end user is. And I think we've had some success. And as Howard was mentioning, just the idea of making someone realize that your patient may not need the dose that you've been giving for years, because your patient is very different than, between patients are different, right? So I think if you start making that building that trust and making the clinicians comfortable with that concept, I think the rest follows a lot more seamlessly.
And I would add that when we talk with patients about whether it's okay for us to do a DNA test or to measure blood levels to guide therapy, most of the time the comment back is something along the lines of, don't you already do that? The idea that we're not personalizing therapy, not individualizing the dose, is concerning to patients. They think that we do that already. And so I've had kind of people say, oh, what do patients think? The patients are often a little bit miffed at us for not doing it sooner, because they know that they're not identical to the person in the next bed. They know that they need something different, and they expect that we would be acting on that.
That's really interesting, yeah, because to see that some patients think it's already happening or should be happening. I didn't consider that. That's a very interesting aspect there. And so in terms of, speaking about the collection of genetic data, that always, brings about considerations and potential concerns about ethical concerns and things around privacy and security. We've been hearing this, ever since those 23andMe and DNA tests and all of that is, and, are patients still wondering where their data is going? And how do you deal with that? Well,
certainly individuals know that when they go grocery shopping, that the grocery store has a lot of information on them, and in many cases sells that information, and there are targeted ads, and there's all sorts of activity that can happen based on that. And they get frustrated that they don't have as much control over who gets it and how it's used and such. The same also occurs with DNA analysis or other types of biomedical assays. But DNA is especially important because a lot of detective TV shows will use DNA to find someone. And there's been famous cases where a DNA test found a relative of someone that was a serial killer and finally found, all sorts of dramatic things that are out there. And so the idea that DNA could be used in some way that is not approved by the patient is a legitimate concern on their part. Within the healthcare system in the United States anyway, and it's also true in Canada and most of Europe, there are protections that are in place around the use of genetic information for guiding care, and the misuse of genetic information for things like denying health insurance in the United States or causing someone to have a different type of health structure in other countries. And so that's protected. But areas like life insurance or long-term care insurance are not covered by at least the US laws. And so we do need to let patients know. Now with pharmacogenomics, where we're just trying to find the right medicine, the right dose, make sure it's safe, there's not a lot of downside to that. We're not finding something that means that you're going to die young or that you're going to be really expensive in a couple of years' time, from a healthcare standpoint. And so there's not a lot of threat there. But the idea that DNA can be used for other purposes definitely needs to be taken into account. And the health system takes it seriously. The research apparatus takes it very seriously. And I think the idea that there are companies that are working with health systems and informatics systems, those all take this, I can't speak for Dr. Roy, but I'm guessing that it's similar with DoseMe. The security of the information is job one. Job one, the reason that there is a job is to get the right care for the patient, the right dose. But the effort that is put out is disproportionate around security, because we just don't want to let the patient down. They've trusted us with information to do good, not evil. And therefore, we need to make sure we're doing good and not evil. And so I think that's a lot of the effort. I don't know, Sharmeen, if you want to add to that.
Yeah, data security and privacy is very, very important. And that's why we at DoseMe, our platform is high trust certified, which is the highest level of certification. We, follow we have to meet all regulatory requirements. And I would say that one of the first questions we get with any health system, that's the first really evaluation that needs to be done in order to take that next step. If the hospital is interested in getting, implementing our system, we have to meet those rigorous requirements. So it's not an option; it's a requirement to even have clinicians utilize our platform for patient care. So I think, just like Howard said, it's not just for the genetic information. In the healthcare space and the dosing, even though it's limited, it's limited to what data is in our platform, but we still have to meet those stringent requirements that are in place, just because of the current, culture and concerns around data privacy with health systems. It's something that, everybody here takes very, very seriously. And it's, again, we wouldn't be able to be utilized by clinicians if we didn't have that in place.
And so speaking of also new technologies and across the board, including in data privacy and security, we're hearing, of course, about artificial intelligence and machine learning just taking over every aspect of our lives. But how can these kinds of technologies help, or are they being introduced in the area of precision medicine? And at DoseMe, Dr. Roy, is that something that you're toying with there?
I feel like every conversation I have,
it's very cliched, but our audiences want to know all about AI.
I appreciate that. I always look at technology as a friend, and so AI is not going away. Machine learning, none of this is going away, and we need to utilize it as an asset rather than run away from it. I think the concern around it is always like, is it going to take my job? Is it going to replace me? But you have to understand, at the end of the day, it's still a clinician, a human taking care of another human, right? So you can't take away from that clinician's judgment. So what the way I approach it and the way we kind of approach, any concern around AI or algorithms, of course, we want to use the data. There's, what it has done is that there's vast amount of data. There's way too much for any one person to be able to quickly process. So that's where AI or machine learning helps us, right? It takes that vast amount of data and quickly analyze it and brings it to the patient level, the care of the patient, where you can then utilize it and make the best decision. So I think that's how we have to look at it. It's an asset. It's not going to, it's sort of complementary to the clinician, right? Or a healthcare system or infrastructure rather than replacing it. That clinical nuance doesn't go away, and only a human can provide that. And so, that's kind of how we look at it. What is there continuous learning that happens with models? Of course, that's how we improve on it and further refine. If you really want to get precise or as precise as is required for that patient, we have to do that continuous learning model and utilize those algorithms. But with that said, at the end of the day, it's still our platform specifically is a clinical decision support software. So the clinician is still the center of that decision making. I would say the difference is, data crunching versus decision making. So the AI part does the data crunching, and the decision making is still made by the human. I don't know, Howard, what your thoughts are on that.
Yeah, I think the applications today are kind of in the middle, where we have rules and data generated by people that are then put into the machine learning or deep learning of strategies. That way, 85% of the lifting in between can be done using informed by human experience, and then a human can finish it off in terms of doing that final consult or whatever is needed. We've learned a lot from a couple of different examples. One is with Watson, IBM Watson. They had IBM Watson Cancer, IBM Watson Genomics, et cetera. And they were very good at getting the data from, let's say, 20,000 feet down to 1,000 feet. A huge improvement, sifting through thousands of articles to give you a stack of articles that you could focus in on that was too much for a human to handle. And so that really taught us that you need to have enough quality data in your models in order to bring it all the way down to sea level, all the way down to the point where action can be taken. And so that's an important thing. Right now I see a lot of AI companies out there, and they're like, oh, our model, from day one, our model will be useful. It hasn't trained in appropriate data, it's trained in insufficient amount of data. The idea that you can build an app as a minimum viable product and that your customers will help make it better is fine if you made Angry Birds, but it's not fine if you are trying to treat someone for a serious disease. And so that's where some of the difference comes in, is there's not the same tolerance of a learning curve in healthcare as there is for the electronic leisure industry type of approach. The other thing is that there have been some very effective models in healthcare. There was a model that looked at skin and would identify the pigmented skin, identify which ones were melanoma and which ones were not. It turns out, and it was very precise, it turns out what it really was doing was detecting whether there was a small little ruler in the background on the skin. Because when you have a cancer, when you diagnose a melanoma, you measure what size it is. And so those pictures had a little ruler in them. And so the model quickly learned, if there's a ruler, it's melanoma, and if there's not a ruler, it's not melanoma. Well, we didn't need a ruler detector. We needed something that could detect. So that data had to be done over again, and it, of course, was not nearly as precise without the ruler. And so, there's things that we're learning. And of course, the last piece I want to mention is around ethnicity. So if you look at work that's being done in a country like the United States or in Canada or many countries nowadays, we have people from all around the world that live in our country and are needing healthcare. And so when you have data that's from one population, such as me, it's useful for people like me. It's not so useful, or not always useful, for other humans. And you think, well, we're all humans. Yes, we are. But there are certain variants that are more often seen based on geography. So if you come from Finland to down to where I am. Yes, you're still a white European-derived individual, but you've come from a different founder of genetic variants that will mean that your care could be influenced in ways that maybe we didn't even measure. Now, with blood level, there's less bias there. But the idea that we use genetics blindly, we need to take that into account, and both for good and for bad. So we can't be saying that someone has a risk of cancer when it's just a variant that we hadn't seen before and is more common in their founder population. In the same way, we can't say that something's a variant of unknown significance when we just didn't do the hard work to prove that it actually caused cancer. So there's still a lot of work to be done within AI to make sure that the kind of data that is training it represents the kind of tasks we want it to perform. And so that's, to me, is the big opportunity.
Wonderful, great insights on AI and why, and keeping rulers out of the picture. So this might be an odd question, but do you think that both of you that we can get too precise? Because it's always important, as we hear about in any field, to also keep the bigger picture in mind. So if we're honing in on, let's say, in grad school, we're really focused in on just one aspect of something just to write a thesis, and we become so concentrated in that. And it turns out maybe we weren't going after the right target in a certain drug development program. So I guess my question has two parts. Number one, can we get too precise, and how do you balance the precision with the bigger picture, whatever that may be, and what, I'd like to hear what that may be from both of you. And number two, how do you select targets? We have so many, targets, and every day there are papers about this is the next big thing. How do you navigate all of that?
Howard, I'll let you start on that one.
Okay, I'll jump in there first. I think that part of the challenge is to define what are we really needing to accomplish, and if it's a precise task, then it will need a level of precision. But most of the time, what we're trying to do is select from amongst equally appearing options. So if you take the treatment of most cancers, we only have a certain amount of cancer drugs that are available to us, whether they're approved by the regulatory authorities or whether they're in clinical trials. There's a finite number of those options. And so often the testing we're doing is trying to put positive weights on some of the options and negative weights on others so that we can more clearly select which therapy should. So it's really more similar to going to a nice restaurant, and you see that there are 7 different entrees, and think, what, I don't really want the pork or the chicken. I'm gonna have the vegetarian option today, or I'm gonna have this other option, and that's the one for me. And so it's all are good options, but which one is the best for that individual? And so that's part of the task is searching from that. And I often use a buffet example. It's not really a very good example because at most buffets you can have more than one entree. But we have a lot of drugs available to us. So can we use one that it has a better probability of success? Do we have one where the toxicity is so severe and is associated with a particular blood level or such that we just want to avoid that at all costs? It becomes no longer worth it to go above a level. Do we have efficacy where we have to get above a certain threshold or else? So I think that most of the examples, and Dr. Roy can talk a lot more detail on this, most of the time we have a target range, not a specific blood level, but a range. And we know that our probability of success is higher when we're in that range. Probability of toxicity might be higher when we're above it, and et cetera. But often what we don't do is we see a new drug come out. We don't really define, well, what is the range for this patient, either at the blood level or just what level of toxicity is acceptable. In oncology, where I spend a lot of my energy, I was trained at a time where you needed to nearly kill the patient in order to kill the cancer. That's no longer true. There is much more precise medicines available that we can now use. How do we make better decisions? And so some of that is old thinking, old regulatory thinking, old drug development thinking that doesn't give us the right kind of data to make a choice post-approval. And so, again, there's a lot of challenges there. Sharmeen?
Yeah, I would say, I'll use antimicrobials as an example, or, a category of medications that have been used with narrow therapeutic index. So that's what Howard was describing, where there's a range you have to be in between to be both be effective, as well as make sure that you minimize the toxicities for the patients, right? And we've learned over time that we're still working on getting it right. So what we thought was, appropriate, even as long as 10 years ago, is no longer really what we should have been doing. So we're constantly learning, and I'll use vancomycin, which is it's probably the most common used antibiotic that's used in the hospital setting. It's been around since the late 50s, and we're still learning how to dose it appropriately. So just in 2020, new guidelines came out that indicated that we should be using AUC guided dosing versus how we were doing it previously, which was checking a level and making sure it's between a trough level that is within a range, and you're good to go. Well, now that's changed, right? And so that's one of the biggest uses of our platform. But that has taught us that what we knew 10, 20, 40 years ago may not be accurate, even though it's a drug that's been around for a long time and it's still being utilized. And so within the antimicrobial therapy, there's a lot of examples of that, where we're constantly learning that, well, if you get to a very precise range for this bacteria, then you'll get better killing. And so I think that's where being precise is very important, right? As we learn that this is the level we need to go to. Now, taking this to the oncology arena, which is Howard's expertise, there are a lot of medications, a lot of chemotherapies that are foundational to treatment. They're not going around, and we just need to make sure that we're learning how to better and more appropriately dose that. And that's where utilizing, a software like DoseMe is very, very helpful, because that's where you can find out that individualized dosing for that patient. So you're not giving them the dose that kills the patient in order to treat them. So that's where it becomes important. So we have to keep the big picture in mind, where there's a lot of variability with the population, but we also need to target specifics, for certain cancers and certain infections that we're trying to use. So, I think it's a balance between the two.
Absolutely. And really being able to more effectively use, I think what I'm gleaning from the both of you is more effectively use existing drugs, right? That for reasons of imprecision may not have been working for patients, which is really exciting. And I think that opens the doors to novel drug combinations as well, when you start optimizing individual drugs and then combinations of them. So it just seems like endless opportunities. So that's really wonderful to learn about. And so I think, where do you see things going in the next, couple of years, 5, 10 years with precision medicine? What is it going to mean? Was it going to mean something different, or are we building on the same idea concepts?
There's a friction in precision medicine between novelty, the hype of novelty, and normalizing precision medicine. And so we see at the same time, we're trying to make something like blood level measurement or DNA analysis a normal part of care, but also talking about the promise of metabolomics or the promise of some sort of functional imaging or these other newer strategies. And that is very much a to be determined type of scenario. Will they be useful? They're definitely cool. Will they be helpful? And really try to define that. And so we're trying to do the same thing at the same time. I was at a board meeting for the Personalized Medicine Coalition on Thursday, and that's one of the things we talked about there was, how do we keep the excitement around precision medicine, personalized medicine out there, so that resources, and I don't mean just dollars, but intellectual resources are spent on solving those problems, but at the same time make it normal. No one gets excited that someone measured bilirubin. It's been a normal part of blood work for decades. It is important. Most people that need to know what to do with it do know what to do with it. And so when you say, oh, bilirubin, nobody goes, oh, bilirubin. Everything that we're doing today needs to become bilirubin, needs to become that normal part of practice. And the idea that we're using something like DoseMe on a 50-year-old drug should cause no one to blink. That should be expected, that we would be using the latest in informational technologies to apply to a drug that might be older than me, but is still very active and effective. And so I think that's part of the friction that we're working with.
Yeah, I think, just like bilirubin or any labs, right? So it's thinking about, just kind of taking a step back and thinking about, when you go to your primary care physician, they do annual lab work, right? So there's a list of lab that they evaluate, make sure your kidneys are working the way they're supposed to, your liver is functioning the way it's supposed to. And we take all that information to do dosing for normal everyday drugs. And so something like pharmacogenomics, that's why I try to how I explain it to providers, that this is just one more piece of information that you should have to take care of a patient, right? So it needs to be normalized in that way, that genomics isn't something scary or something only, a specialized group of people can utilize. While there are experts, but it needs to be part of the normal care of the patient, because, that patient, just like you're looking at their, renal function to figure out what's the best drug for them, or their, vital signs or their other lab values, genomics needs to be just that common and that normalized, that it's incorporated when you're making those decisions. And I think that's kind of where we need to get to. How we get there, I think it's an effort. It's an effort from all of us. It's, making sure we're not ignoring the cool things that are coming, but also keeping in mind that those really important factors, and at least dosing and, picking the right dose for the patient or individualizing dosing, those are already available. And we just need to make everybody comfortable with using it on an everyday basis.
Well, thank you to the both of you for your efforts in the field and for making these things happen and for normalizing precision medicine for patients. So thank you so much for the work you do. And thank you, Dr. Roy and Dr. McLeod, for being on the show today.
Pleasure. Thank you.
And sharing your insights with us.
Yeah, thank you.
Thank you.
All right, that's the end of this episode of the X Talks Life Science podcast. We hope you enjoyed the show. If you like today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next time. Bye for now.
To join the X Talks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.